Hello, my name is Jason Samarasena, and I'm an interventional gastroenterologist at the University of California, Irvine. And I'd like to take a few minutes today to talk about our research study entitled EUS-guided in vivo imaging of the porcine esophageal enteric nervous system using needle-based confocal laser endomicroscopy. Now, before I get started, I'd like to thank and acknowledge the co-authors on this work, Dr. Andrei Chanowski, Dr. Amrita Alawalia, Dr. Susuma Shonora, Dr. Keedon Choi, Dr. John Lee, and Dr. Kenneth Chang. A little background about our study. The GI tract is innervated by the autonomic enteric nervous system, which is mainly composed of the Meissner's plexus and the myenteric Auerbach's plexus. The Meissner's plexus is found mainly within the submucosa, while the Auerbach's plexus is located between the circular and longitudinal muscle layers of the muscularis propria. The enteric nervous system is thought to have an essential role in GI motility, regulation of blood flow and secretion, and other functions. And many diseases like gastroparesis, esophageal dysmotility disorders are thought to be a result of a dysfunction in the enteric nervous system. And one of the major challenges with studying the enteric nervous system thus far has been visualizing it. And right now, this is mainly based on full-thickness pathologic evaluation. Endoscopic visualization has been difficult. But a study by Oya and colleagues published in GIE in 2012 demonstrated in vivo imaging of the porcine esophageal enteric nervous system using probe-based confocal laser endomicroscopy, the CellVisio system by Mauna Kea Technologies. Since the probe-based system has a limited depth of imaging penetration, they needed to access the nerves using submucosal endoscopy with an invasive flap valve mucosal technique. Also in that study, peristaltic movements, contact bleeding, and entrapped air bubbles also hampered the imaging. Now recently, a novel ultra-thin confocal laser endomicroscopy probe has been developed by Mauna Kea Technologies with a 0.85 millimeter outer diameter, and this probe can be passed through the bore of a 19-gauge FNA needle. So the main aim of our study was to visualize the enteric nervous system in porcine esophagus in vivo using endoscopic ultrasound-guided needle-based confocal laser endomicroscopy and a local fluorescent neuronal probe neurotrace. An additional goal for this study was to evaluate the feasibility of ex vivo imaging of the esophageal neuronal network from biopsy samples obtained at the time of endoscopy using a novel through-the-needle forceps biopsy technique. For our methods, 4 Yorkshire pigs were examined under general anesthesia. In each animal, imaging was performed in the mid and distal esophagus. At each site, imaging controls with saline were performed prior to any imaging with neurotrace. For the procedure, a linear echo endoscope was advanced to the esophagus. A 19-gauge FNA needle was used to inject 5 milliliters of normal saline into the submucosal layer and to the interface between the circular and longitudinal muscle layers. After saline injection, the NCLE probe was advanced through the FNA needle, and imaging within the submucosa and between the MP layers were performed by directing the FNA needle under endoscopic ultrasound guidance, and then gently advanced the NCLE probe out of the needle tip. Now for our actual experimental imaging, we use the fluorescence neural probe neurotrace. With the needle still within the same site in the esophagus, 5 milliliters of neurotrace was injected into the previously targeted submucosal and muscularis propria layers. The NCLE catheter probe was gently advanced out of the needle tip and imaging was acquired. The time to visualize the neuronal structures was recorded, and the quality of the imaging was graded on a scale between 1 and 5. Now, for the ex vivo experiment, submucosal tissue biopsies from the porcine esophagus were obtained for ex vivo evaluation based on our recently reported technique, where we took a 19-gauge FNA needle and inserted it between the submucosa and the MP layers, and a miniaturized cold forceps was then passed through the 19-gauge needle and advanced into the submucosal layer. Under EUS guidance, the forceps was opened up, advanced slightly, and then closed to obtain a biopsy specimen. Now these specimens were then immediately placed in a petri dish in a dilute concentration of a neurotrace for 10 minutes. The probe-based confocal laser endomicroscopy probe was then gently placed in contact with the tissue sample in the petri dish, and imaging was performed. Our results showed that imaging of the esophageal enteric nervous system using endoscopic ultrasound-guided needle-based confocal laser endomicroscopy was successful, both within the submucosa as well as between the muscularis propria layers, and clearly visualized neuronal cells, glial cells, nerve fibers, and bundles, providing their distinctive imaging patterns. The NCLA visualization of neural elements started at about 30 seconds after neurotrace was injected, and neural elements were visualized in all examined esophageal sites. Image quality of the neural elements was excellent with a mean score of about 4.7 out of 5. Now, the forceps through the needle technique was successful in acquiring pieces of tissue about 3 to 4 millimeters in length, and after incubation of the samples in neurotrace, the probe-based confocal laser endomicroscopy system was clearly able to visualize ex vivo the neuronal elements in the tissue. In fact, the neuronal elements were very similar to those obtained in vivo with a needle-based confocal laser endomicroscopy system. In summary, our study shows that using the needle-based confocal laser endomicroscopy probe under endoscopic, ultrasound, and fine needle guidance, in vivo imaging of the esophageal enteric nervous system could be achieved without mucosal dissection or resection. In fact, with the needle firmly secured in the tissue, the imaging was excellent. There was no bleeding or tissue injury present in the resected specimens, and the imaging was not affected by peristalsis or entrapped air bubbles. The time required to image at each site in the distal and mid-esophagus was relatively short. As a result, potentially multiple sites of the enteric nervous system in the esophagus could be evaluated with this method in a relatively short amount of time. Limitations of this study include the small sample size. Neurotrace for human use is currently limited as the human safety of neurotrace injection is unknown. However, our ex vivo through the needle forceps technique may serve as a potential alternative approach, as we were successfully able to achieve images similar to those acquired in vivo while circumventing these toxicity issues. In conclusion, our study demonstrates that EUS-guided in vivo imaging of the enteric nervous system is feasible without mucosal resection and also provides a novel ex vivo imaging alternative to humans. These minimally invasive imaging approaches could potentially better characterize diseases of the GI tract such as echalasia, esophageal dysmotility disorders, gastroparesis, and possibly provide insight into diseases associated with visceral hypersensitivity, such as irritable bowel syndrome. Thank you very much.